All right, welcome everyone. So today we're going to be talking about epigenomics and using that as a way to introduce also hidden Markov models, which is one type of sequential learning model that we kind of alluded to last time. So what is epigenomics? Epigenomics basically fits within this first module. Basically, first we talked about expression analysis. Basically, how do we understand gene expression patterns? And this is fundamental in many places. Then we talked about, and we used that to introduce clustering classification, Gaussian mixture models, etc. Then we talked about some of the specifics of single cell analysis and how do we understand single cell data in a much more sort of foundational way. The problem set is very much addressing both epigenomics and single cell and expression analysis, etc. Who has already started working on the problem set? Please raise your hands. Okay, awesome. So please show up at recitation tomorrow. First, take a look at it. And two, go to recitation tomorrow. Tomorrow in recitation, we're going to spend the whole hour going through P-set 1. Okay, so this is, again, there to help you learn. It's there to teach. It's not there to make it hard for you. So basically, there's a lot of numbers of steps that we want you to go through for P-set 1. Some of these steps are complicated, so we're going to be walking through them together. 2 questions, okay?
Yeah,
3?
tomorrow's a student holiday.
Oh, gosh. Yeah, well, okay. So, I know the answer, but the question is, how does a tenured professor get kicked out of MIT? And the question is recitation on a holiday. So we're going to basically do office hours instead, and we're probably going to just record it so that people can't make those office hours are able to do so. 2 options for office hours. 1, we could do it later today so that you guys have the recording over the weekend. Another possibility is that we can do it on Monday. Are you guys free? My little girl had her birthday, so everybody like, are you 1? Are you 2? Are you 3? Who's free at 3? Who's free at 4? Who's free at 5? Are you 6? Okay, so who's free at 4 today? Who's free at 5 today? It can be over Zoom. You don't have to come over. Who's free at 6 today? Who's free at 7 today? Who's free at 8 today? Who's free at 9 today? Who's free at midnight tomorrow? I'm kidding. So it doesn't seem like a lot of people are free today. When would you guys like to do it? Do you want to do it on Monday? Monday's good? Oh, yeah. I see a lot of nodding for Monday. Good. Who's free at 3 on Monday? Good. Awesome. Who's free at 4 on Monday? Nice. Who's free at 5 on Monday? 6 on Monday? That's amazing. All right. So we're probably going to do it at 3 on Monday or 4 on Monday. Who's free at 4 but not at 3? Who's free at 3 but not at 4? It's a tight call, 3 versus 2. All right, so we're going to send a doodle poll, and it's either going to be at 3 or at 4 or at 5 on Monday. Sounds good? So try to work on it over the weekend. Take a look. Ask ChatGPT. They're your friend. Don't ask for the answer. Ask for you to understand the code. Basically, go through stuff. Basically, our goal is for you to learn and to sort of, empower you to do all that stuff. Okay? Great. So that's for the P-set. And next week, next week we're going to be talking about regulatory genomics and regulatory networks. But today we're diving into epigenomics. So what is epigenomics? Epigenomics is the ability to sort of have a diversity of cell types from the same underlying genome. So first we're going to look at what is epigenomics, overview, what are chromatin modifications. We're going to talk about the technologies on the experimental side. Then we're going to talk about the primary data processing. Basically, when you get the signal out, what do you do? We're going to introduce a super cool Burrows-Wheeler transform approach that was introduced by Bowtie for very rapid gene mapping. Yesterday, I had a wonderful time hanging out with Langmead, the original author of the Bowtie algorithm, who was basically visiting MIT. So we're going to talk about read mapping and peak calling. Then we're going to turn to not just primary data processing of one epigenomic mark, but how do we combine multiple epigenomic marks together to characterize chromatin states. And this is going to start with foundations of hidden Markov models. Who has seen hidden markov models before? Who has never seen hidden markov models before? Okay, good. And we're going to see how that applies to a multivariate hidden markov model. Then we're going to talk a lot about model complexity. How do you decide just how many parameters, how many states does your model have? And then how to learn chromatin states jointly, not just across multiple marks, but also multiple marks in multiple cell types. Everybody with me here? Awesome. Let's dive right in. So epigenomics. Epigenomics is basically what allows you to go from a single genome to the extraordinary diversity of cell types we have in our body. Basically, if you look at the brain, there's an extraordinary diversity within even the neocortex. But if you look at subcortical regions, the number and the diversity of neuronal and both excitatory and inhibitory neuron subtypes is just enormous. If you look at our immune system, our blood cells are, of course, there's red blood cells, but then there's like these, huge diversity of white blood cells and all of the functions that they play in the immune system. If you look at just any little snippet of your skin, just within it, there's, hair follicles, there's innervations, there's sensory organs, there's, epithelial cells, there's, veins and blood irrigation, etc. This is all coming from the same underlying genome. All of these cells have exactly the same code. So what allows them to have such extraordinary diversity in their phenotypes? The fact that instead of just having one code, you basically have different subsets that are utilized by each cell type. And the way that this is enabled is through epigenomics. So very briefly, your DNA is packaged up extremely compactly within every one of your cells. If you take the DNA from one of your cells, it is 2 meters long end to end. It's extraordinary. 2 meters worth of DNA in every one of your cells. And you have like 1 trillion cells. So if you put now 1 trillion times 2 meters, you don't just get to the moon, you get to Jupiter, I think about 10 times. Okay, so this amount of DNA compacted within your body is, and within every one of your cells, is relying on structural mechanisms of epigenomics, whereby the DNA is looped around these nucleosomes. So nucleosomes are about 250 nucleotides worth of DNA. Or let's say 200, it's about 144 for 2 loops of the DNA around the nucleosome and another 50 nucleotide linker. So basically 200 base pair chunks are labeled in different way. So that's because every nucleosome can have decorations of these proteins that it's made out of that we call histone proteins. And these histone proteins have markings that tell the cell whether this is an active region, that it should be actively opening, whether that's a repressed region, whether that's a poised region, whether that's a transcribed region, if it's a promoter, an enhancer, and so on and so forth. Is everybody with me here? I saw your hand go up. Okay, so basically there's 8 histone proteins in every one of your nucleosomes. And these nucleosomes capture about 200 bases of DNA. So that plays a structural role in compacting this, massive 2 meters worth of DNA into a tiny chromosome, or at least 23 pairs of those. And it also plays a functional role in basically remembering which regions are important for every cell type. So at every cell division during your development, there are factors that establish the epigenomic memory of every one of your cells to be able to remember its state as you go on through your life, so that your neurons don't wake up tomorrow thinking that they are, immune cells. Everybody with me here? Awesome. So basically DNA is very long, cells very small, huge compression. To use the DNA, you have to actually open up this structure. And that's why you have to remember which structures you're going to be needing in every one of the cell types. And then DNA accessibility, the state of the chromatin, and there's a huge role of DNA interactions across different segments. Everybody with me here? Awesome. So now we talked about the fact that DNA packaging is both structural and functional. There's 3 ways to encode this functional part. So first of all, the structural compaction has a functional implication that the more compact it is, the harder it is to open. So these regions in your neurons that are super highly compacted are probably not helpful in neurons. They're helpful in heart or in liver or in lung, or you name it. Everybody can hear? Yeah. So one of them is just the compaction. The second one is accessibility. Basically, compaction is one thing of the entire fiber. And the second one is locally, where are the nucleosomes positioned? Can they sort of roll back and forth to, even in a loosely compacted region of the DNA, of the chromosome, be able to locally open it up for regulators to bind that piece of DNA. Is everybody with me here? Good. So basically, one of them is just DNA accessibility. Another one is DNA methylation. So on the DNA sequence itself, we have A, C, G, T, but you can put a methyl group on top of a C. So it becomes a methyl C. Now, a regulator, a transcription factor, when it binds DNA, it doesn't just open it up to read it. What it does is that it binds from the side. And if there's a methyl group there and you're feeling the T base or the A base or the G base or the C base, when you're feeling and you're looking for a C, if there's a methyl group, you might not recognize it. Everybody with me here? So therefore, by modifying the C, a regulator might no longer be able to bind. And conversely, by modifying the C, a regulator that was previously unable to bind might now be able to bind only the methylated version of C. Is everybody in here? So then that means that I can actually change the code of the DNA. I can change the recognition, the way that regulators are reading DNA, by adding a methyl group directly there. Everybody with me here? So that's one type of modification. One type of modification is DNA methylation. The second one is compactness and accessibility. And then the third one is based on directly these histone proteins that I mentioned earlier, where every nucleosome is made out of 8 histone proteins. The most common version is H2A, 2 copies, H2B, 2 copies, H3, 2 copies, H4, 2 copies. Okay? So basically, every nucleosome has H2A, H2B, H3, H4, each in 2 copies. And you can have now modified nucleosomes themselves. For example, instead of H2A, you might have a variant, H2AZ, or you name it. You can have modified H3, modified H4, but you can also have post-translational modifications on the tails of the histone proteins themselves. So basically, the histone proteins are providing a way for DNA to attach, but they're also hanging out with loose tails that can be recognized by specific regulators that are then attracted to whatever signal you put into that tail. And that signal can basically say, oh, well, this is a region that you should use as an enhancer. This is a region that you should use as a promoter. This is a transcribed region. This is a repressed region, etc. Is everybody with me here? So there's methylation that changes whether a TF can bind or not, a transcription factor can bind or not. There's compactness that basically tells you if that region at all is accessible. And if within the accessible region, I can actually bind those nucleotides. And then there's histone modifications, which are modifying the tails of these proteins with read signals. And there's other write factors that will then come in and change these modifications that will then guide how that region is interpreted. Everybody with me here? Now, some of these modifications don't change the properties of the histone itself. So you can add a methyl group, 2 methyl group, 3 methyl group. It still has the same properties, like biophysical properties. But if you add acetylation, that actually loosens up the DNA by itself. So some of these modifications are not just having informational impact. Some of them are actually having a biophysical impact by actually changing the compactness, the accessibility, the looseness, the breathing of the chromatin. Who feels that they've learned something? Yes? Awesome. Great. 5, 4, 3, 2, 1. Good. Who's with me? 5, 4, 3, 2, 1. Okay. Awesome. Beautiful. Any questions? Yeah, please.
Oxidation and carbon methylation are lab generated, not lab generated, but they're probably used for the DNA exposed in the lab.
Sorry, there was an opening of a bottle exactly as you were asking. Something is generated, lab generated?
So oxidation and carbon methylation are PTMs that are commonly.
Oh, yeah. So basically, I spoke about DNA methylation and DNA acetylation that are the most common. Now there's ubiquitination, there's sumoylation, there's all kinds of additional ones, including some man-made ones. So basically, you can use that to mark up regions, but then you need to maintain them because every time the DNA is replicated, those are going to be lost. So you need to have regulators that establish them and read them and rewrite them and so on and so forth. So again, part of the purpose or part of the point of the double helix is that it's semi-conservative replication. So basically that means that I'm copying it and I now have each copy can basically serve as the template for the other. But now the trick is, yes, it can serve as a template for the DNA sequence, but what about the epigenomics? I need to have the right regulators that will basically read one copy and then reestablish that copy. And now, as you start thinking about sort of these histone marks that are on top of the DNA, when you undergo replication, sometimes they just fall off. Other times they fall off one way and then they're reattached right behind the replication fork. Same thing with RNA transcription. To transcribe, you need to open up the strands. Something's going to happen to these histones. So basically, there are some marks that are transcription associated. There's some marks that are co-transcriptional. Along with the transcription machinery, these are carried along, and then they're modified, and so on and so forth. So you got to think about both the informational view of the genome, but also the biophysical view of the genome. Other questions? Yeah.
Yeah, that can be very interesting, but how it relates to the last lecture. So we started lecture. On last lecture we were looking at like natural mutations.
Yeah.
So here we see like sort of like artificial mutations or.
Yeah. So many people think about epigenetics, and I try to keep saying epigenomics. Now, genetics has a connotation of inheritance, and therefore epigenetics is very often associated with inheritance. And basically people say, oh, this is not a mutation, it's an epimutation. So basically when an organism, for example, undergoes some kind of exposure to extreme cold or to radiation or to all kinds of things, maybe the DNA will change, but sometimes the epigenome will change. And now the question is, are these epigenomic modifications epigenetically inherited? And then the answer there is nearly all of the time, no. In other words, humans go out of their way to wipe out the epigenomic state from one generation to the next. When the sperm is formed and when the egg is formed, whatever modifications we had basically are very sperm-like and very egg-like. Now, I get an epigenomic modification in my brain. How's that going to be reflected in my sperm? Not clear. Now, when the zygote forms, there's a wiping out of the epigenomic information to basically restart clean with a bunch of primary sequence signals that are sitting in the DNA itself. And of course, the egg itself has all kinds of proteins that are establishing that signal. So this is part of the reason for and then the zygote itself, re-wipes like several times in sort of during its development. So if I have an epigenomic modification because I saw, I don't know, a violent movie yesterday, I have to somehow pass it into my sperm. That sperm has to make it into the egg with that memory somehow that traveled from the brain cell to the sperm cell. It has to make it through the egg, and on the zygote, it has to now sort of carry out the modification. And then after I have fully developed, my child has fully developed, that kid has now must have some kind of epigenomic modification in their brain and obviously in all of their organs. So it's complicated. It's difficult. Now I've just hopefully convinced you it's possible, but let me now tell you how it might be possible. It might be possible that small RNAs that are associated with epigenome are basically traveling throughout the body, making into the germline, making into the next generation, making it through the, milieu of the egg cell, And somehow, as the embryo starts developing, you can basically now have these small RNAs continue to sort of carry out epigenomic modifications. It's possible, but very, very difficult. Any other questions? Yeah.
How's the this the course from a mechanistic standpoint, how does it know this shape to think depending on
So the compactness is established as a memory by regulators in the previous cell type that you were at. So basically, the zygote forms, you have one cell type because you have one cell. Then there's the first cell division. One of these cells is going to become more anterior, the other one will become more posterior. The anterior cell, in some species, gets marked randomly. In other species, based on the direction in which it sits inside the mom or inside the egg or inside, the whatever environment this is replicating. So other species establish gradients of expression for different regulators. So you basically have an anterior-posterior gradient. Then you have two additional gradients, one of them going down, one of them, going down one way, one another way. And there are some response elements that require 2/3 of one and 1/3 of the other to respond. And others, 3/5 and 2/5, and so on and so forth. And that basically can now establish the expression of additional regulatory signals that are, specifically in one band of that gradient. One stripe, the way that they call them in the Drosophila element, for example. Or in C. elegans, every replication is programmed. You know exactly the identity of that cell is like 13A, it's going to replicate into 27B and 27D, and that one is going to replicate exactly this way, etc. So there's many, many different ways in which evolution has kind of established this symmetry breaking, if you wish, to sort of establish all of the different cell types. In my view, it is miraculous and extraordinarily beautiful how this happens. I mean, I challenge any engineer to basically create a robot that's sort of self-replicating and sort of then deciding on the fly with 3 billion letters of code how to, form itself and sort of this extraordinary innervation I showed you for 1 millimeter of skin. I think this is, this probably took billions of years of evolution. That's why it took so long to go from unicellular organisms to multicellular, because you have to establish these rules of, self-reprogramming and differentiation. Any other questions? Who finds the questions really enlightening, right? I mean, ask what you're wondering about, because again, I can give you the lecture, I can give you sort of answers to the stuff that you're burning to know. Any other burning questions? Okay, great. 5, 4, 3, 2, 1, who's with me so far? Yay, awesome, great. So that last one I mentioned is kind of cool. Basically, this one is about compactness. This one is about, changing the TFs that bind you. This one can have both a biophysical role and a read-write role. And this one is by far the most complex, the most versatile. You can have, in these histone tails that I mentioned before, amino acids that are modified with different types of modifications. So you can basically, we're going to be talking a lot about H3K4 trimethylation. What the heck does that mean? H3 means that from all of the proteins, so there's H2A, H2B, H3, H4, from all of the proteins, it's histone H3. Everybody here? Then which residue? So it's lysine, which is K, at position 4, 1, 2, 3, 4, or lysine at position 36, 1, 2, 3, 4, all the way to 36. Everybody with me here? And then what do you add to it? You can have methylation, you can have phosphorylation, you can have a ubiquitination, you can have simulation, you name it. And you can have one methyl group, or you can have, HCH3, and one of the H's becomes another CH3 and another CH3, etc. Okay? So you can have methyl, 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 also known as trimethyl. So when I talk about H3, you're going to be like, oh, histone H3, no problem. K4, oh, lysine 4, no problem. Me3, oh yeah, 3-methyl, no problem. Everybody with me here? Awesome. So now each of these will now be associated with particular functions, but they're not going to be acting alone. H3K4ME3 coupled with some of the modifications means one thing, but coupled with a different one means a different thing. Okay? So in addition to that, as I mentioned, you have DNA modifications. You can have methyl C in CpG. You have nucleosome positioning, DNA accessibility, etc. And you can think of all of that as a constant struggle, a constant fight for territory. On one hand, the regulators are trying to bind DNA, and they're sort of pushing nucleosomes apart to bind there. Some of these regulators are stronger than others. We call them pioneer factors. They're able to sort of bind DNA and displace the nucleosomes. Others are obeying whatever the nucleosomes say, basically say, oh, you're not binding here. It's like, no problem, I'm not binding there. Everybody with me here? So there's histone, proteins. There's nucleosomes, which are made out of histone proteins. There's general factors, there's chromatin regulators, and so on and so forth. Okay? And all of these are basically competing for access to the DNA. Okay? So there's a biophysical component, and then there's a read-write component where specific proteins will come and recognize these modifications and then do stuff like loosening up the chromatin, like tightening it up, like bringing a bunch of their friends and sort of establishing a landing site for RNA polymerase to start, to bind and start transcribing and so on and so forth. Everybody with me here? Awesome. Good. And as I mentioned, they don't act alone. So for example, in enhancers, you have DNA accessibility, but you also have H3K27 acetylation. That basically means an acetylation group in the 27th lysine of histone H3, or H3K4ME1, promoters H3K4ME3, and more K9 acetylation. For transcribe regions, 3 marks, K36, K79, K20, H4K20, ME1, and these are actually found at different places along the gene body. At the beginning, in the middle, and the end of transcription, basically have slightly different abundance of those. And then there's 3 types of repression. There's H3K27 trimethyl, which is polycomb facultative repression. So basically, in a given cell type, this is sort of shutting down or opening up things. H3K9 trimethylation, this is a very stable heterochromatin repression. This is like the super, super compact stuff that I mentioned earlier, where when you're there, you basically are not getting out. And then there's DNA methylation, which is very orthogonal, and it's usually a mark of repression in regulatory regions. In transcribed regions, interestingly, more methylation is associated with more expression, which might simply mean do not bind here to reinitiate. So it might actually still be a repressive mark, but a consequence of transcription, and please don't reinitiate here. Okay? Everybody with me here? Good, yeah.
What actually are the UTRs?
Untranslated regions are basically when you transcribe an mRNA, you have the start of transcription, and then you have the start of translation, which is the ATG. Between transcription start and the ATG, that's a UTR. It's the 5' UTR. And now between the TAG, TGA, TAA translation stop codon and the end of transcription, that's another UTR. It's the 3' UTR. So after you basically make your transcript, you add the cap as a way to stabilize it. And after you're done transcribing, you add the poly A tail to also stabilize. Sure. Other questions? All right. Awesome. So again, hundreds of known modifications, many still emerging. We need a systematic mapping with chromatin immunoprecipitation, bisulfite sequencing, and DNase accessibility. So now let's talk about these technologies. So basically, bisulfite sequencing basically means that you treat the DNA with bisulfite, and that will differentially alter Cs that do not have a methyl group and Cs that have a methyl group. And then you sequence the results twice. And based on whether you see a C or whatever letter a methyl C looks like, then you're going to know, oh, this is methylated or not. Everybody with me here? So that's bisulfite sequencing. Then DNase sequencing basically means I'm going to randomly chop up the DNA and find the segments that were just not bound by anything, or that were So basically, how am I going to randomly chop out? By adding DNase. It's the enzyme that digests DNA, but does so preferentially in open chromatin regions and accessible regions, the regions that are not marked by histone modifications. Everybody with me here? It's kind of interesting because DNase will have a very funny shape on the DNA itself. So DNase will basically cut in accessible regions, but for a region to be accessible, you need to have a regulator that makes it accessible and that pushes off nucleosomes. So you'll basically see an accessible region, but right in the middle of it, there's an inaccessible region. That's exactly where the regulator is binding. Everybody with me here? So that's basically DNase sequencing. And then chromatin immunoprecipitation, we're going to talk a little bit more, but it's basically finding an antibody that will recognize these, pull them down, and then you're sequencing the chunks of DNA that came with whatever your antibody bound. And these chunks are basically the places where you had each of these modifications. Okay? So that's chromatin immunoprecipitation. Immuno means you're using an antibody to pull down. Precipitation means pull down. And then chromatin. Okay? All right. So there's a lot of efforts, for example, the Epigenomic Roadmap, the ENCODE project, and others that are basically mapping many, many different tissues in the body and histone modifications, open chromatin, DNA methylation, and gene expression. Okay? Why? Because there isn't just one human epigenome. Yeah, there's like one human genome that each of us has. And, you know, with some exceptions, of course. But there's many, many, many epigenomes that each of us carries. My brain epigenome might be different than my lung epigenome, and also within my brain, my neuronal epigenome might be different than my, you know, inhibitory neuron or oligodendrocyte epigenome, etc. Everybody with me here? So every cell type and every tissue, and frankly, every stage of development, sometimes every person, males versus females, will have different marks. And, you know, we basically need to figure them all out. Okay. All right, so to do that, there's basically many different projects, ENCODE, Roadmap Epigenomics, Blueprint, Mouse ENCODE, Mod ENCODE, etc. And, you know, they're mapping many modifications, many cell types, many individuals, many species, many conditions, many antibodies across the whole genome, etc., etc., etc. Okay. As I mentioned earlier, the way that we do chromatin immunoprecipitation is by building an antibody that will basically recognize a histone modification. So you basically are feeding, you know, some unsuspecting bunnies a bunch of H3K4ME3, and then, you know, their immune system is like, oh gosh, what's that thing? I'm going to build a bunch of antibodies against it. You just collect the antibodies, and now you are able to pull it down. Everybody with me here? Same thing with transcription factor. You can kind of bind, instead of the modification, you can bind a regulator. And you can find all of the regions where the regulator is bound. So you basically do this experiment. You pull down. So you first chop up the DNA, then you pull down all of the regions that were bound by either, you know, this antibody here or that antibody there that recognize this modification or the binding of this regulator. And then you get rid of the nucleosomes and you sequence whatever segment of the DNA came along. Sounds good? Doesn't matter how you sequence them, but then you map them to the DNA. You map them to the genome sequence, and then you're like, oh, well, in this region here, I pull down H3K4ME1, you know, much more here than here. So that tells you that this region of the genome had that modification. Everybody with me here? And same for this line here, H3K4ME3, H3K4ME1, H3K36 trimethylation. So H3K4ME1 is a mark of enhancers. So you basically see that there's these enhancers here saying, oh, there's some active region. Then there's a promoter, and then there's a transcribed region. It's kind of cool. You can just read it off. That's basically the general accessibility of the establishment of these control regions. Then you have the promoter itself. That's exactly where transcription starts. And then you have transcribed regions, which are along the body of the gene. And guess what? The gene model is right there. There's the start codon, there's the transcription start, there's the translation start, there's the translation end. These are the UTRs, the untranslated regions on both sides. And then you have a transcription stop. And you can basically see that the exact place where transcription starts has basically this H3K4ME3 signal. And guess what? There's a dip here. Why is it absent? It's absent because that's exactly where transcription starts. So you basically need to bind that place, and guess what? There's not going to be any histones there. There's not going to be any nucleosomes. So you can't have a histone modification if you don't have histones in the first place. Everybody with me here? Cool. So again, always think about the informational and the biophysical side by side. Okay? So this is for 3 marks. You can do this for dozens of marks. You can basically have DNA accessibility. You can have, various promoter-associated marks, various enhancer-associated marks, active enhancers, repressed, polycomb repressed regions, K27 trimethyl, heterochromatin, DNA methylation, and, we're going to talk at some point about high C, which is chromatin conformation capture. It basically tells you about long-range interactions. And in addition to all of the marks, you have now transcribed regions that you can see have transcription-associated marks, but also RNA sequencing. And then you have the actual gene models at the top. So the question is, how do we make sense of all of that? How do we summarize these multiple marks into chromatin states? How do you learn the combination of all of these things to effectively understand the language of the epigenome? Everybody with me here? Awesome. Good. So ultimately, what we want to know is what are enhancers, promoters, transcribed regions, repressed regions. Promoters are basically the regulatory regions that surround the transcription start site. Transcribed regions, repressed regions, you understand. Enhancers are slightly different than promoters. Enhancers or promoters are basically the names that we give the control regions that are right at the start site or a little further away. And they have different properties. Promoters are typically marked whether or not the gene is actually transcribed. They're kind of ready for business. Enhancers are extremely cell type specific and tissue specific. So the enhancer will be on in, very specific conditions, whereas the promoter is like, hey, just come to me when you're ready. Everybody with me here? Now, every gene will typically have 1 or maybe 2 or 3 promoters. That's sort of the places where you start transcription. But a gene will typically have 20 or 50 enhancers. So basically, there's many, many different places where this gene will be used. So you're going to have an enhancer for your left toe, an enhancer for your right toe, and an enhancer for your, left ear, and so on and so forth. Everybody with me here? I mean, yes, I'm making this up. Like we're actually bilateral animals, so usually your left toe and your right toe will be the same. Okay. Cool. Learning stuff. All right. Who wants to see some math, algorithms? Good. Let's dive in. So now, what's the problem we have? We now have a gazillion reads that we just got. How am I going to map these reads to the genome? I just sequenced 100,000 reads from each of 1,000 experiments. So I have 100 million reads to map to the genome. How are you going to do it? Raise your hands. You guys know. You saw it on Tuesday. Yeah. Alignment. Great. What are different ways I can do alignment? We saw dynamic programming, right? Alignment could be an exponential problem. And we turned it into quadratic. We were so proud of ourselves. Then quadratic was a little too slow because I now have 100 million reads that I want to map. And I have now 3 billion bases for each of those reads. Let's say each read is 30 nucleotides. I have 30 times a billion times 100 million. That's a lot of bases. That's a lot of mapping. That's a lot of alignment. So what could we do instead? What was the end of the lecture? Yeah. K-mer hashing. Good. So instead of basically saying, oh, I'm just going to construct a dynamic programming matrix, I'm not looking for evolutionary changes here. I don't care to find a very nice, beautiful path with a bunch of mutations. No, if it doesn't match, it doesn't match. So I'm going to basically super rapidly hash all of the K-mers and then just map them to a hashed version of the genome. Everybody with me here? Good. This is very powerful. But if I want to hash every, 10-mer, and I want to compare it to the 3 billion letters of the genome, I'm going to have a very big table with, 3 billion bases times all of the hashes. Everybody with me here? So basically, that's actually quite expensive to basically have that long list. So what we're going to see today is a crazy, super awesome algorithm that Ben Langmead actually developed, who was here visiting me yesterday. And I told him, hey, tomorrow I'm teaching about bowtie, to basically map reads super, super fast on the genome. And it might just blow your mind a little bit, but we kind of like that. Excited about a little bit of mind blowing? Yes? All right, let's blow a little bit of minds. So how do we map millions of short reads to the genome? We're going to look at traditional hashing schemes and then the Burrows-Wheeler transform that was implemented into the bowtie, which of course has BWT. All right, quick exercise to the reader. Just map CA, GGG, TC, etc. Here. Who finds it first? Raise your hands. Good. As you all saw, it was here. Sorry, you're laughing because it was so simple. So yeah, last time we're like, oh, how many matches do you have? Now I'm like, okay, just find it. It's there. Just find it really fast. So how do you find it really fast? Well, you could basically allow mismatches. You want to allow mismatches. You want to allow sequencing errors. And you want to be super memory efficient. So you want that entire operation to be executed super, super fast without sort of constantly trying to swap in and out the genome from memory, okay? So you can basically do sequence alignment, you can do hashing, you can find other advanced algorithms, for example, linear time string matching or suffix trees and suffix arrays. The challenge is that the memory requirement is typically quadratic for those. And the Burrows-Wheeler transform is actually order M. It's super, super memory efficient, super, super fast. And it has been the new norm since Ben Langmead basically developed bowtie, okay? You can basically here see Burrows-Wheeler transform. I asked how many citations he has. I think he said something like 90,000 citations for this, like, you know, bioinformatics paper. What is it? Like, it was published in Genome Biology. The impact factor of that journal has probably gone up 4 times, like by 4 or 5 points just from this one paper, just because it's insane. So it was basically saying, oh, we have this, you know, cute new indexing scheme that we found from, you know, BZIP2, and we just applied it to genomes, and it basically makes it in practice 35 times faster than the previous algorithm and 300 times faster than another algorithm. Now, if you want to spend 300 hours or 1 hour aligning all your reads, which one are you going to choose? Okay, so that's exactly what it's about. And right after this, you basically have SOAP that became SOAP 2. And then, you know, this algorithm became, you know, the version 2, which always sort of incorporated bowtie. So how does it work? Okay, brace yourselves. This is very fun. So you have a bunch of chromosomes, and you can put them end to end, and you can extract seeds and, you know, create this giant table. And you can basically take a short read and then hash it and then map each of these hashes and then look up where that table falls. It sounds super fast, but it's very memory expensive, okay? Instead, what you can do, and it sounds crazy, but bear with me, is you can take up your genome and just chop it up into little segments and sort all of these little segments based on an algorithm that I'm going to say, that I'm going to describe very shortly. And I'm basically going to shuffle my genome in a way that preserves the ordering of specific bases. And I'm going to explain what that means in a second. But what that allows me to do is now take a new short read and then start mapping first one letter, then another letter, then another letter, then another letter. And progressively, I'm searching for all of the T's. All of the T's are just at the end of the genome because I've sorted it. But then all of the ATs are somewhere else in the genome in a smaller interval that I can kind of use to look for next. And the AATs are in a yet smaller interval. So it's a form of beam searching where you're starting with a very large beam and then you're making smaller and smaller and smaller and smaller every time until you find a stretch of your shuffled up genome that contains all of your matches to basically any substrate. Sounds a little crazy, but again, bear with me for a second. So the first thing that we're going to do is basically figure out what the heck is this Burrows-Wheeler transform. So Burrows-Wheeler transform was basically introduced in, I think, BZIP2 as a way to compact files. If you have a file that has a bunch of, you know, consecutive letters, for example, the genome has like, you know, a bunch of A's and a bunch of T's and a bunch of C's, etc. You basically say, oh, well, there's 30 A's in a row and 20 B's in a row, etc. Okay. or 20 Gs in a row. So it basically started out as a transformation that allows you to then compress things really fast and really tight. Okay. What does that compression look like? It basically takes any word like banana and it looks at all of the possible rotations of the word banana. Okay. I'm going to put a start character, an end character, in every possible rotation. Everybody with me here? Now I'm going to sort all of these rotations. So basically, you know, alpha numeric is alpha, you know, lexicographically. So I'm going to take all of the A's, put them first, all of the B's, N's, you know, start and stop. So far, so good. And now the Burrows-Wheeler transform is going to be the last column of that string. And that last column of that string turns out to have some nice properties about the stretches of similar letters, stretches of nucleotides. So far, so good. Now, you can basically now say, okay, what the heck did you do to my genome? I want my banana back. And the answer is no problem, I'll get you your banana. Okay, how am I going to get the banana back? First, I'm going to take the last column, which I know is the last column, right? How can I get the first column if I have the last column? Raise your hands. I have one answer. I have the last column. I want to find the first column. What's special about the first column? I have two answers. What's special about the first column? Three answers. Go ahead. It's sorted. So what can I do to the last column to find the first column? Sorted. Okay, I'm good. So I get my something back and I get, oh gosh, I have the sorted version back. Okay, well, that's a little better. But now, what's the trick? The trick is that if I have the last column and the first column, because they're all the rotations, I basically get the first two columns. Because if I put the last column and the first column together, this is the second column, basically. And I sort it, I have my second column. Is everybody with me here? Because the last column and the first column are basically consecutive, if I have the last column, I can get the first column. And if I have the first and the last column together, I can basically put them together and I sort them. And that gives me the second column. And I can figure out the third column. How? By appending the first column and the second column to the last column and resorting the whole thing. Everybody with me here? So basically I do last, first, second, sort, I get third. Last, first, second, third, sort, I get fourth. Last, first, second, third, fourth, sort, I get fifth. Give me a 5, 4, 3, 2, 1 who's following. Awesome, good. So when I'm done with the whole thing, I can get back my full matrix, and therefore I can find simply the character that says start, and then that's my full string. Okay, everybody here? So basically, I don't need to remember anything but the last column to be able to reconstruct the whole gene. Okay, so this was fun, but it didn't get us anywhere yet. So, you know, it still doesn't tell us how we're going to find something. And I apologize, I'm not as self-centered. This slide was actually made by Jason Ernst when I asked him to give a guest lecture. So he chose, I don't know, some random name with a bunch of repetitions. And my parents would be so proud. So basically, if I want to find the string OLIS in this gibberish stuff here, okay, how do I do it? Remember this beam searching approach. I'm going to start with an S, and I'm going to have a beam for all of the S's. And where are all the S's? Well, in the first column, they're just at the end. So far, so good? So all I need to remember is how many characters do I have of each type? So that's one variable. The count of how many characters occur before C lexicographically in the gene. Okay? That basically simply tells me where the string of S's will come. So far, so good? So I have one pointer. Then, if I add the I is not that far from the S. It is just before it. And because all of these are cycles, just before it is basically right there. Now, not all of the S's will be I's. So basically, my beam is going to get smaller. Everybody with me here? So all I need is
variable. I have the query substring, of course. Then I have a variable that basically tells me how many characters occur in before C lexicographically in the genome. And then the most interesting variable is the occurrences of character C in the far right column, starting at the current position SP. Now, what's SP and EP? It's the start pointer and the end pointer. So as I'm doing my beam search, I have a start pointing and an end pointer. And at every iteration, I just keep updating the start and the end pointer so that I keep switching from column to column, basically, from first to last, from first to last, from first to last, every time I add one more character. Because as I add that one more character, I've now gone from, oh, okay, well, I had IS, and now I have LIS. And I have LIS, and I add one more, and I have OLIS, etc. Everybody with me here? So basically what I'm doing is every single time I have a start pointing and an end pointer, the start pointer is simply wherever I start, like the number of counts of before character C in my genome. And then the end pointer is just simply that plus one. Okay, basically the next character plus one. So if I have, the S's start here and say the T's start there, then that's my start and my end character. So that's all of the S's. And then I progressively keep making it smaller because I can use this pointer, table here, that basically the number of occurrences of character C in the far right column starting at that position. Okay, and then building that can also be done very, very fast. Okay, and now I basically go from the S to the IS, from the IS to the LIS, and then suddenly it starts decreasing in length because only a subset of those characters will actually be in the next one line over. Okay, so this is extremely fast because I only use one string effectively, the last column. The first column is implicit. I don't need the first column. Do I need the first column? No. It's just the occurrences matrix. This C matrix is basically the first column. It just tells you, well, you have 300 A's and then 400 G's and then, 200 T's, etc. Is everybody with me here? So the first column is basically sorted, so I don't need it, but you can think of it as having it. And that allows you to keep going from the last column to the first column. And then the occurrences table basically tells you about that last column. And then you're just updating your pointers. And in the end, you end up with all of the locations in your genome that have that exact substring. Why is that exciting? Because I can basically now do it 100,000 times, get all of the coordinates, take the 3 billion letter table, map out how many times did I end up at this position in this letter here, which is a unique position of the genome. And the reads are now piling up in that unique position. And what do I do at the end? I just reshuffle my genome to rebuild the original one while preserving the counts. Everybody with me here? Awesome. So that's the key idea of the Burrows-Wheeler transform and bow tie in sort of mapping these reads to the genome. Okay? So it's very little memory usage, basically same as the input or actually less. You don't represent the matrix of the strings, just pointers. And in the encoding step, you simply sort the pointers. In the decoding, you follow the pointers. The original application was string comparison, so bzip2. Runs of letters were compressed into letter run length. And in bioinformatics, it's about similar runtime at hash tables, but dramatically faster in practice because of memory efficiency. Okay? And you can then map 100,000 reads, only transform once. So you pre-process once, you reverse transform once, but then after you've mapped all of the counts, you have. Who thinks this is kind of cool? Yeah? Rate 5, 4, 3, 2, 1. Who's with me? Yes? Awesome. I see some 3s, some 4s. You guys are honest. This is great. Yeah. Expand the acronym. So basically the way that it works with mismatches is you could artificially put in the mismatches and just search that string a bunch of times. You could basically say, oh, I want to look for that string or one of here, one of here, one of here. Yeah, I think that, that would be sufficiently fast. I'm sure you can do some, even faster things. Any other questions? Okay, good. So we've now mapped all our reads to the genome. And we want to start inferring now, not just where are all of the reads. So basically what we've ended up with is something like this. It's an intensity. So what we've achieved is what we had promised, namely, I took 100,000 reads and I've now mapped them, and I have these heights here. Everybody with me here? The next step is, what do I do with that? Are these reads distributed in a way that suggests that the experiment was successful? Are they matching across different marks in ways that allow me to infer more information about the genome? So how are we going to do that? We're going to basically run a bunch of quality control steps, understanding that this will allow us to figure out which experiments were good, which experiments were less good, and so on and so forth. Okay? You don't have to worry about all of those, you know, but this is useful for your general education. So basically, first of all, you need to know where are the regions that are bound by a regulator. But the problem is that when I chop up the DNA to basically figure out where are the regions bound by a regulator, I will also get the regions that are just generally accessible. So I might think naively that the regions that are generally accessible are also bound by the regulator. So what I need to do is the experiment twice. Once with the antibody to pull down the regulator, but also once with just DNA by itself. In other words, DNA and all of its accessible regions. Is everybody with me here? And then the ratio between the two tells me which regions are truly bound by the regulator. So that's step number one. Step number two is, you know, as I'm sequencing, sometimes the quality of the reads diminishes. So, you know, I don't want to catch all of those. Section number three is what fraction of the short reads were actually fully mapped, uniquely mapped, or multiply mapped. Okay, so you can, you know, deal with that as well. Another one is about how the reads are actually matching. If the reads are simply piling on top of each other perfectly, then this is likely to be an artifact, because that's probably just one RNA molecule or one DNA molecule that was sequenced over and over and over and over again. Because if you chop up the genome randomly, you're going to get a bunch of different starting points. So another one is basically, what is the non-redundant fraction of binding of my library? Everybody with me here? Another one is the fact that every time I pull down a regulator or a histone modification mark, I'm going to have some breaking up of the genome. I'm going to pull down all of the reads that were attached to that segment that I pulled down, and I'm going to sequence the endpoints of those fragments, because I only sequence from the beginning of a DNA fragment. So now you have to think about the fact that I'm capturing fragments of the genome, and then I'm sequencing either from here or from there into that fragment. Is everybody with me here? So if I pull down 500 base pair fragments, then I'm going to have reads that are pointing to the right anywhere within, I don't know, 250, 300, 400 base pairs of what I pulled down. Suppose that the regulator was binding, I don't know, 50 or 100 nucleotides. Okay. Now, or you know, this histone mark is perhaps covering 144 nucleotides. All of the reads, the 200 base pair segments that contain that mark that was pulled down, are going to start to the left of that. And all the ones that I'm sequencing from that side are going to start to the right of that. Is everybody with me here? So then I'm going to have some kind of characteristic left-right pointing read distribution. If I read from the left, it's going to look like the Watson strand. If I read from the right, it's going to look like the Crick strand. So if I have a read on the Watson strand pointing to the right, I'm going to have to assume that it's somewhere to the left of where it was bound. And the ones from the right are going to be, you know, on the Crick strand are going to be to the right. Is everybody with me here? Can I get a 5, 4, 3, 2, 1? How you're following this? Awesome. Bunch of 5s, some 4s, some 3s. Okay. So because there's a physical segment of the DNA that I'm capturing, and that physical segment better be pulled down by my experiment, that segment better be 500 base pairs long. Am I going to get any reads starting here and pointing to the right? No, because that segment would not have been pulled down by my experiment. So all of the ones that were pulled down are basically the ones that are starting to the left. And all the ones from the right side are the ones that are starting from the right. Okay? So that basically allows me to do one more quality control step. I can basically look at the distribution of forward and reverse pointing reads and scan them past each other. And when they meet, I should get a peak. And when they don't meet again, I should get a trout. So I can basically look at the self-correlation of the forward reads and the reverse reads at varying offsets. And at those varying offsets, they will be matching at some point, and that will basically tell me what was my fragment length. Everybody with me here? Awesome. So now that fragment length better be high enough compared to my read length. And the read length simply says, how much did I read and how much did I read? And therefore, if I have just random fragments, I'm going to get a bunch of stuff at the read length, okay? And those reads are going to be amplifying each other, and therefore, that will be a sign of a bad experiment. So basically, if I have a bunch of peaks here, it just simply means that I keep resequencing the same reads. If I have something beautiful there, it basically means that, this is a good experiment, okay? So that's the fragment length distribution. All right, so now I can reject the bad experiments and start doing some, peak calling. I want to know where are the regions that are bound, okay? So I have a continuous signal, and I want to figure out intervals where that signal was bound. Is everybody with me here? Awesome. So that basically means that I need some kind of model for how are reads distributed if I have a binding event on the genome. And I can basically say, okay, up to 15 reads, I might not say it's significant, but at 150 reads, I'm going to start believing. So there's many, many different methods that have been developed to recognize that. For example, Max and Peak-Seq have different properties based on whether they're window-based, whether they cluster the tags, whether they subtract the background signal, whether they calculate a false discovery rate, whether they have some kind of normalized control, and so on and so forth, okay? But most of it is going to be about what is the probability of having some count, given some kind of local expectation, and some kind of Poisson distribution based on those, okay? And the challenge is going to be, okay, at what threshold do I call a reasonable peak? And those thresholds are going to be based on, just the some number of reads. So then the great challenge is going to be, how do I select the real peaks versus the bogus peaks? And here's two replicates of the same experiment. The same experiment was carried out twice. And in replicate 1, I see, some agreement that looks nice. And then in replicate 2, I see some agreement. But now I see this giant peak here and only a smaller peak here. And I see a moderate peak here and a very, very small peak here. How do I combine these replicates? There's different ways of doing it. One of them is take only the peaks that were called in both experiments. That means that this one is going to get rejected because it wasn't perhaps also called there. Another one is, let's add up the signal from the two experiments. Seems reasonable. The problem there is that you're wasting the effort that you did to sort of carry out two experiments. If one of them is bad, you're just adding a bunch of noise. Another one might be, I'm going to take the union of all of the peaks. If I called it here, and if I called it there, and if I called it there. So basically, I'm going to take all of the ones, like this one was called here, that one was called there. That sounds great. Take the union. The problem again is that if one of them is noisy, you're just, you're going to call all the noisy ones. If you take the intersection, if one of them didn't have enough signal, then you're not going to call all of these great signals from the other one. Okay? Good. So I hope I've convinced you that we need to do something more than that. The something more than that is very simple. It basically says, let me sort all of my peaks in experiment number 1 and see how many of those replicate in the other experiment. And based on the replication rate, I'm going to choose where to choose the cutoff in experiment 1. Everybody with me here? And I'm going to do the same for experiment 2. In experiment 2, I'm going to sort all of the reads based on their intensity. And I'm going to go down the list and say, oh, wow, this replicates well. Oh, this stops replicating. Why is that powerful? Because the replicates well could be 10% to 2% transition, or 50% to 30% transition, right? So the moment I stop replicating well, even if one experiment was much less signal than the other experiment, I'll still see that the replication rate decreases at some point, but that point can be very different for experiment 1 and experiment 2. Everybody with me here? So basically, I go down the list of experiment 1 and peaks, and then I stop. And maybe for experiment 1, I'll choose like 4,000 peaks, and for experiment 2, I'll choose like 300 peaks. And that's okay. Sounds good? But if a peak was so strong in experiment 2, in the 300 peaks of the experiment 2, if it was so strong, I'll still keep it, even if it may have been missed in experiment 1. Okay, can I get a 5, 4, 3, 2, 1 as to who's with me here? Awesome. Nice, nice. I see a bunch of 5s. That's great. And some 4s. Okay, so that's the key idea. Basically, I say, what is the replication rate, if you wish? And then this replication rate will, you know, or this non-replication rate will basically start, you know, going really bad at some point, and then that's where I'll choose my cut. Okay, so that's sort of the key idea of this replication rate. All right. Now, what have I done? I've basically carried out my experiments. I've mapped my reads. I've called my peaks. I've replicated them. The next step is, how do I now combine multiple signals from multiple marks? Okay. So here's the challenge. We have dozens of histone modification marks. They can arise in very complex combinations. They're extremely diverse, extremely dynamic. And maybe there's distinct functions for distinct combinations of marks. And there might be both additive and combinatorial effects. How do we find the biologically relevant combinations of marks? What we want is to be able to learn the language of the epigenome from scratch. We want an unsupervised approach. Just give it a bunch of data, come back and say, okay, well, I found 20 patterns that matter. Everybody with me here? So that's the goal. And we want a probabilistic model that explicitly models the combinations and that uses, just like we saw on Tuesday, the relationship between neighboring positions to avoid making a bunch of bogus scrolls. So what we're going to do is basically try to define this based on the fact that serially, as I walk through my genome, I will go through different combinations, and I want to infer the hidden chromatin state. Okay, so I have a bunch of observations of individual marks, and I have a hidden chromatin state track. At every position, I'm in a enhancer state, promoter state, transcribe state, etc. And I'm emitting a vector of histone modification marks. Okay, so I want to have both a transition matrix that basically tells me that promoter states are near promoter states, enhancer states are near enhancer states, transcribe states are near transcribe states, and emission that basically tells me promoter states are more likely to emit these marks, enhancer states are more likely to emit those marks, you know, repressed states are more likely to emit those marks, etc. Okay, and to do this, we're basically gonna develop a hidden Markov model. Okay, again, who has seen hidden Markov models? Raise your hands high. Who has not seen them? Raise your hands high. Okay, awesome. So what are hidden Markov models? Hidden Markov models are basically a way to start thinking about the type of sequence that generated something. Okay, for example, in speech, a hidden Markov model can be used to transcribe the words that I'm saying based on the utterances that you hear. Why? Because these utterances come in a sequence, they come in an order, and the one before can influence the way that you interpret the one right after. Is everybody with me here? Yeah. So the time here is the position in the genome from left to right. Because if I'm in an intergenic region, I'm more likely to stay in an intergenic region. If I start, say, an exon, I'm more likely to stay in a gene, and so on and so forth. So I'm capturing the temporality as simply position along the genome. And I'm using the neighborhood information to constrain the extraordinary number of possibilities for any one segment. Everybody with me here? Awesome. So what we want with this hidden Markov model is the ability to both emit a DNA sequence of a certain type, to recognize DNA sequences of that type, and to learn the distinguishing characteristics of a given state. Everybody with me here? Awesome. Remember at the very beginning of the class, I basically said that there's a model and a bunch of observations. The observations are the stuff that we see about the world, and the model is the stuff that we infer about the world. And I showed you these as being basically independent, even though they were sampled from a continuous process that has some kind of temporality, okay? And then we talked about how in the generation, we have the probability of observing the data given the hypothesis, and in the inference, we have the probability of being in the spring state given the observations that I have. Everybody in here? Awesome. So now, what I'm going to do is I'm going to add this temporality component. I'm going to basically say, well, a transition from, promoter to enhancer to transcribed, etc. A transition from summer to fall to winter, etc. And I basically have some notion of the state that I'm in that depends on the previous state that I was in, right before. Okay? So that's a Markov chain. And this basically says after, this particular observation, I'm more likely to see that observation. So in a Markov chain, everything observed, whereas in a hidden Markov model, the Markov chain is not between the observations, it is instead between the hidden states. And the hidden states generate observations in a generative framework and transition from one state to the other. Okay? So I'm going to have two sets of parameters. I'm going to have my observations, x, my hidden state, pi, and the two sets of parameters are going to be what is the probability of transitioning from this state to that state. And that probability is not going to depend on anything. Okay? Given the state that I'm in, I randomly draw an observation and I randomly choose a next state to go to. This is what we call memorylessness. The model has no memory. It's memoryless. Everybody with me here? So if I'm in this state, I have some generation probability and I have some transition probability. The transition probability is simply the probability of going to state L given that I was in state K. The emission probability is the probability of emitting character Xi given that I'm in state K. Okay? So we have a vector of observations, a hidden path, a transition matrix, and an emission vector. Okay? And then we're going to be using the same Bayes rule as before, but now we're going to have not only emission probabilities, we're also going to have transition probabilities from the states nearby. Everybody with me here? Awesome. So this is an example hidden Markov model for interpreting genomes. So basically you have some background distribution of nucleotides, equal probability for every A, C, G, or T. And for example, a GC-rich sequence might have higher probability of emitting a G or a C with 40% compared to only 10% for an A or a T. Everybody with me here? So I have some self-transition probability that basically says if I'm in a background, I'm more likely to stay in a background for a long time. 100 times out of, 99 times of 100, I'm going to stay where I am. And then here, 95 times of 100, I'm going to stay where I am. So 19 times out of 20. But one time over 100, I'm going to transition from background to promoter. And one times out of 20, I'm going to transition from promoter back to background. Which state do you think is going to be longer? This has a very high self-transition probability. Background is probably going to be longer. Everybody with me here? Awesome. So now I have an observation. I basically see, ATT, AGG, TCTA. What is the chance that this was generated by background only? Well, 0.25, 0.25, 0.25, 0.25, and of course the transition probabilities, because there's a 0.01 chance of exiting every single time. Everybody with me here? So that's the joint probability of observing this sequence and all background. And that is 0.25 to the 10th, because that's the emission probability of each of the characters, and then 0.99 to the 9th, which is astronomically small, 10 to the minus 9. Very, very small. Why is it so small? Because I could have generated many different types of sequences. This is just one of basically an exponential number of sequences. Because at every position, I have 4 options, times 4, times 4, times 4. So 4 to the 10. Everybody with me here? Awesome. Now, if I generate exactly the same sequence, but from the promoter state, then the chance is 0.1 for all of the AT-rich sequences, and then 0.4 for all of the GC-rich sequences. And therefore, that gives me 2 to the 10 times 2 minus 9. And now I can compare the two and say, aha, this one is 2, 10 to the minus 9, and this is 5. So this is 2.5 times more likely. Okay? And therefore, this sequence, as you can see, is more AT-rich, so it's more likely to be from the background state than the promoter state. Sounds good? Can I do better? Maybe I could just transition from the B to the P to the B. So from the background to the promoter, back to background. Why? Because here I capture all of the AT-rich, here I capture all of the AT-rich, and here I capture all of the GC-rich. Who thinks this is going to be better? Raise your hands. Who thinks it's going to be worse? Raise your hands. Why? Good, good. Because transitioning is extremely expensive. Okay? So if I compare the two, now I'm 10 to the minus 10. And it's much less likely because of the high cost of transitions. Okay? All of that is nice and good. And all I need to do is just simply calculate every possible parse and just evaluate them all, and I'm done. Happy? How many parses are there? Exponential. Okay? So I need to do something better. Yeah. Yeah, you could basically say which state am I more likely to start in. You could also say, you know what, I don't want to start state probability. What I want is just simply the, continuous, basically, if I'm more likely to self-transition with background than with promoter, then I'll just expect more background. So I could just get rid of this as a parameter. Okay, so I'm not going to score them all. Instead, I'm going to use dynamic programming to turn an exponential number of possible paths to a polynomial. How? Effectively by aligning states to positions. So it's the same thing as we did on Tuesday, where I aligned a sequence to a sequence, but now I'm going to be aligning states to positions. Is everybody with me here? But the alignment of states is not going to transition linearly through all of the states. It's going to jump state to state according to the transition probability matrix. So going from this state to that state has some probability. Going from that state to the next state has some other probability. So I'm going to be aligning these states with reuse, and the transitions I'm going to be paying for according to the transition matrix. Can I get a 5, 4, 3, 2, 1? Who's with me? Nice. Yes. So as you're transitioning, you're not paying the same fixed gap penalty. You're paying a penalty according to that specific transition. And every transition is going to have its own parameter. Okay? So I'm basically going to encode this as I've already computed the best possible score of parsing that entire sequence all the way to this state. And that gives me a score for every one of the previous positions. And I'm going to use that to compute the best possible score for each of these next states based on the score that I got all the way to that position, the transition probability penalty of going from there to here, and the emission of generating the next character. Okay? So my next maximum is going to be the emission from the current position. And then I'm going to choose the maximum of all of those options. What do I want? I want something that has a great previous score. But I don't want to pay a huge transition penalty. I want something that has a great transition score. But I don't want to start from a crappy previous state. So it's going to be the product of those two that will basically tell me what the current maximum is. Can I get a 5, 4, 3, 2, 1 who's following here? Nice, awesome, beautiful. Now, one more thing. I make that maximum decision. What do I need to do? Remember it. So I'm going to store both the next maximum and the arrow from where it came from. Everybody with me here? So then I get to the end, and then I trace back, following back the arrows. At every position, I choose not from 3 previous squares, but from a whole column of previous squares. I have kn entries to fill in. And at every position, I choose any one of k previous steps. So this gives me a k square n time and only a kn space. Everybody with me here? Who feels that they've learned stuff? Yes? Awesome. So now that I have that, I want to also perhaps calculate the total probability of generating a sequence. What is that total probability? It's the sum of all of these probabilities. So then I can sort of propagate that all the way to the end. And then I can do something very fun, which is if I observe a particular sequence, both before and after, I can calculate how likely is it that I parse all of these and end up in the state B, coming from the left to the right, or how likely is it that I parse all of these starting from state B, and I calculate that from the right to the left. And if I add the two together, what that gives me is the total probability of going into a particular state and out of that particular state, okay? So that is the total probability of being in a state given all of the stuff before and all of the stuff after, okay? So what that allows me to do is to now use all of that information to effectively create a hidden Markov model that allows me to parse the probability of being in each of these hidden states of enhancer, promoter, transcribe, repressed, based on the observations of the marks that I'm emitting. Is everybody with me here? Awesome. So what have we seen in today's lecture? We basically saw, the foundations of epigenomics. We saw about primary data processing, about mapping of reads using the Burrows-Wheeler transform. We looked at various ways of doing quality control and peak calling. And we looked at some foundations of hidden Markov models for generating sequences and for parsing sequences and for decoding sequences. We haven't yet looked about how do we learn the parameters of a hidden Markov model. And we still need to talk a little bit about model complexity, learning chromatin states, etc. All of that is not in your homeworks. So we're going to go pick up this lecture on Tuesday, and then we're going to continue with the regulatory motif lecture. And the two are kind of tied with each other, okay? And then for hidden Markov models in particular, we saw the basics of observations versus models, of using Bayes' rule. We looked at the difference between Markov chains, where I'm just transitioning between different observations, and hidden Markov models, where I'm transitioning between hidden states and I emit observations. And then we looked at the Viterbi algorithm, the forward algorithm, a little bit of the posterior decoding algorithm that basically allows me to align effectively a set of hidden states to a set of observations. Who feels that they've learned stuff today? Awesome. Good. Have a wonderful long weekend. See you guys on Monday for recitation, either at 3, 4, or 5 p.m. We're going to send out a poll, and we're going to go through P-set 1 then. And then we'll continue this on Tuesday next week. Thank you.